Should we start, guys? Okay, let's start. Let's get going. So today we'll talk one of the very interesting stuff in kind of tackling DNA damage or response, that is termed as SOS response. And mainly it has been seen in the case of first time it was seen in response against DNA damaging agents. So this will be a part of the continuation of the DNA repair we have discussed earlier. When I looked at the syllabus, the SOS response was not covered yet. So I thought it might be worthwhile to spend, and it is one of very interesting subject. So SOS response or SOS repair response was first described or proposed by Miroslav Radman in 1974. It has been quite some time now. It has been more than 50 years now. But yes, we will be discussing today further. Down the discussion, you will come to know SOS response. Still, it's very relevant and very, very interesting at the present moment. How SOS response came about is some of the observations that was seen by other researchers, as well as Miroslav Radman himself. Two key experiments or two observations what has come forward for the proposal of the SOS repair system is that, first, if you look at the organisms, for example, in around the time people were looking in 1974, it's all UV radiation, and look at the response to it and try to find out enzymes which are responsible for the toxicity or for the rescue from the UV based toxicity. So what it has been observed at that time is that even if you give high dose of UV, many a times the E. coli cells or the host cells are very much able to survive. So then there should be some mechanisms which can be involved in rescuing or making the cells survive at high dose of UV radiation. And another observation, what it has been seen is that in certain circumstances, the E. coli could afford to give pyrimidine dimers. That is the result of UV radiation exposure. So the experiment here, what we are looking at in this slide, is the survival of phage. Phage are the viruses for the bacteria. As you can look at it here, when there is no UV exposure, the survival of the phage is around 1.6 into 10 to the power 4 cells. Okay, herein, what do you mean by that is that if you look at here, the phage survival is calculated, or in other words, the phage which is being looked for its survival is already exposed to UV, and look at its survival. So when the host cell is not radiated, there is no radiation of the host cell, the survival of the UV exposed phage is 1.6 into 10 to the power 4. If you look, if the host cell is exposed for 5 seconds for a very short time, which gives a survival of the host cell 98%. In this case, what you can see very observing is that what is important is that the survival of the phage improves 2.1. So what does it mean by this? If you look further down, 15 seconds, 30 seconds, 60 seconds, in this exposure, the survival of the host cell goes down, but the survival of the UV radiated phage improves. What it leads to the hypothesis is that inside the host cell, when it is exposed to UV radiation, it induces certain kind of response which helps in better survival of the phage. So the Radman, when he looked at this, what his hypothesized was that the first thing, when you give lot of UV radiation or a long exposure to UV, the cell, rather than induces or leads to the expression induction of certain genes, which leads to better survival of the damaged product, damaged DNA. So what he looked at is that he looked at the mutations of certain genes. For example, UVRA, okay, that is the repair gene. Okay, repair gene, as we discussed in earlier cases, UVRA is a repair factor. If you have a mutation in repair enzymes, what happens is that the phage survival drastically drops as you give exposure to UV different doses. Again, the DNA polymerase mutant again behaves very much similar to the ones wherein repair is defective. So the earlier observation in the case of wild type, in fact, the phage survival in the wild type is much better. What does it mean? The observation here, what he has seen is that the UV exposed phage survive better when the host cell is exposed to UV radiation. And this better survival links to DNA polymerase or DNA replication and the DNA repair. That's how Radman comes about the proposal that there is a repair system which kicks in for better survival upon DNA damaging agents. So there is a inducing, of course, we discussed earlier, there is a basal DNA repair system exists, but when it goes really high dose, there is a system wherein a DNA repair system is being induced to make the cells survive better inside the system. So that he called or he proposed to be SOS repair. So SOS, I think, is more of there is no expanded form of the SOS, but I guess it comes from the Save Our Soul, but there is no evidence of it in the paper why SOS, the full form of SOS. So now, before going into that understanding, when we discuss about DNA repair or homologous recombination repair, what did we say is one of the steps which is involved in homologous recombination repair is that there is a single-stranded region which is which helps in the polymerization of RAC, and RAC helps in finding the homologous pair and continues about the continues for recombination repair. What is to be noted here is the fact that Reg-A forms a filament in these single-stranded DNA regions. This has to be noted down. Okay, and another uh, effect is that what we see is that during bacterial conjugation, when there is a gene transfer from one bacteria to another through pilus, uh, the organelle. So what happens is that the basically it has been studied in the F plasmid of E. coli, and during this transfer you can see here single strand DNA is only transferred, which is converted into double stranded DNA in the uh, acceptor cell. Okay, donor cell has a double stranded. Again, you have to note down here that there is a single stranded DNA involvement here. One, there is a single strand DNA involvement in the repair, and then single strand DNA involved in during conjugation. This will have relevance later on. And again, when we are looking at a replication, again, if there is a DNA damage, the replication fork can stall, and uh, there can be expose of the single-stranded DNA regions during replication. Okay, when there is a stalling. So this helps in the polymerization of Reg-A in this single-stranded region. Why we have been discussing so far about single-stranded DNA and the polymerization of Reg-A, it is very, very related to SOS response. How? When there is a single-stranded DNA, the Reg-A forms a polymer or filament on the single-strand DNA. This single-strand DNA, when Reg-A binds and forms a filament, what is interesting here is that the Reg-A polymer allows the binding of or forms the binding site of a protein called LEX-A. So why this binding inter subunit here? You can see one subunit of Reg-A and another subunit of Reg-A in that polymer forms the binding site of LEX-A. So when there is a DNA replication fork stalling or the homologous um, recombination repair, wherein you have the Reg-A polymer formation, or conjugation where the single-stranded DNA, which can be recognized by Reg-A and can form filament of Reg-A, it allows the formation of a binding site of a protein called LEX-A. So why it is important? This LEX-A, once it binds to these filament of Reg-A, the Reg-A filament activates the autoproteolytic cleavage of LEX-A. So it cleaves, the LEX-A is cleaved into two, and it's no more able to bind to DNA, unlike the full-length LEX-A. So LEX-A full length is a DNA binding protein. It binds and does its activity. Meanwhile, when there is a single-stranded DNA regions, Reg-A filament formation allows the binding of free LEX-A to this filament of Reg-A, thereby activating the autoproteolytic activity of the LEX-A, giving rise to two products which is not able to bind to the target DNA sites. So what is coming here? So far we have been talking about many stuff. Single-stranded DNA can also be involved in homologous recombination, or there can be ways to indicate. So here is the point. the SOS response that we have been talking about and which was proposed by Ratman in 1974, what it has come to known is that it involves REC-A filament formation and it is acting through LEX-A. Okay, so how LEX-A is involved? That is very important to understand SOS response. So, okay, now REC-A is very important for the LEX-A function, in that LEX-A is a repressor. Okay, when it is in the full length, LEX-A binds to the promoter region and doesn't allow the expression of downstream genes, and all these downstream genes are SOS response elements, and these SOS genes, which are generally repressed by LEX-A, involves in DNA repair, damage tolerance, and then brings in cell cycle progression checkpoints. Now, as we were talking about, when there is a single-stranded DNA, REC-A forms a filament and leads to the cleavage of LEX-A. The cleaved products no longer can inhibit the or repress the transcription of SOH genes. Now, when people looked at the chromatin immunoprecipitation experiments using LEX-A, where does it binds, they could figure out there are around 30 genes which have LEX-A binding consensus sequence sites, and another 19 sites wherein it doesn't have the consensus LEX-A motif. So overall, around 50 genes are regulated by LEX-A directly or involving the consensus sequence site or unrelated sites as well. So now, if you think the SOS response, which is carried about or bring or induced by REC-A through LEX-A, has a very complex network to repair the DNA damage, and meanwhile you stall the progression of cell cycle progression so that the cell has enough time for the damaged DNA can be taken care, or you can synthesize or replicate even in the presence of damaged DNA, which would slow down the replication process. So this process integrate the process. This response is called the SOS response. Now, some of the target genes, two of the target genes, SEL-A and UMU-DC. These are two genes which has LEX-A binding consensus sequence, and what it has been seen is that the SEL-A or UMU-DC are induced only at a very high level of DNA damage point. So why these genes which have consensus LEX-A sequences respond or induce very late? So the explanation is that the consensus sequences allow LEX-A binding very tightly at those sequences. So if you think about it, when there is a damage, there will be single-stranded DNA because of the stalling of the replication fork due to the damage, or enhanced level of DNA repair, something like homologous recombination or other recombination repairs, wherein there is exposure of single-stranded DNA. the level of lex A keeps on decreasing. So to make sure that the cell cannot cope up, the lex A has to come down to very, very low level for cell A and UMUDC promoters to be cleared from the lex A repression. So the binding sites, this makes this ensures that the SOS response are induced when there is a heavy dose of DNA damage. So it is kind of when at a very uh, when there is a lot of DNA damage and the cell doesn't have any other way to cope up, then only the SOS response are induced. Okay, so now that we were talking about the stalling of replication fork, that is likely to be the first thing to happen when there is a DNA damage, and that leads to the exposure of single-stranded DNA regions, which allows the formation of REC-A filament, and REC-A filament leads to the cleavage of LEX-A and induce SOS response, which induce cell cycle arrest or induction of the DNA damage repair enzymes. And of course, other way in general scenario when there is not much DNA damage, still it can continue for the replication fork to be restarted that we discussed earlier, the chicken feed uh, solution that we discussed earlier that can kick in here when there is not much high dose of DNA damage. So what is very interesting very recently is the involvement of antibiotic resistance and the SOS response. Anyone? Okay, let's continue. So why it is very, very important is that what it has been seen is that if you have low dose of antibiotics, in fact, it increases the antibiotic resistance. And how that is done is through um, independent of so one of the fact is that the uh, resistance to or the uh, when you have a lot of these things going on, SOS response is coming up. One of the target genes is like the peptidoglycan synthesis proteins. So that would affect the integrity of uh, the membrane of the bacteria or the cell wall component of the bacteria. So even in E. coli, what we talked about is that there are two lipid bilayer membrane, and there is a very thin layer of peptidoglycan. So when SOS response kicks in, the peptidoglycan synthesis or the integrity of this lipid bilayer is disrupted. So then, how does the cell respond to the this kind of response or threat to the integrity of the cell membrane or cell wall component is that there is something called two-component system. What it does is that it involves two components. One is a kinase, histidine kinase. Okay, in this case, the CPXA. Okay, and that when there is this disruption in the peptidoglycan or protein lipid bilayer, it activates the histidine kinase that phosphorylates the one domain of itself, the cytoplasmic domain of itself. It's an autophosphorylation. Okay, so this autophosphorylation, there is a phosphorylase system. I think we have talked this earlier also. Phosphorylase system transfers the phosphate to CPXR in this case, that goes and act on certain uh, reasons. So many of the SOS response leads to the disruption of the cell wall and cell membrane, induces cell the synthesis of other proteins like protein folding degree, efflux pump. All these are affected, and in even the SOS response is also induced. Why it is very, very important here, the SOS and two component system, is that this feedback system in many of the bacteria, what it does is that there is something called the there is pathogenicity islands, wherein, what do you mean by pathogenicity islands? There is a part in the genome wherein the pathogenesis related genes are located. So what happens many a times is that when SOS response kicks in, the pathogenicity island in many of the bacteria can come together and package itself and forms a particle, something like the bacteriophages. Okay, it can package itself as a like bacteriophage, and this can help in spreading pathogenicity inside the system. So SOS response, if you induce it with low dose of antibiotics, what it helps is that rather than curing the pathogenicity, it may help in spreading the pathogenicity. Okay, so SOS response has a lot of links to antibiotic resistance or pathogenicity itself. And how does it link to antibiotic resistance? Many of the antibiotic resistance genes are encoded in phage genome. For example, tetracycline resistant gene, chloramphenicol resistant gene, many of these have come from the phages. So these phages, many a times, okay. So many a times, what happens is that these antibiotic resistant genes are present in bacteriophages. Yeah, these bacteriophages, what happens is that when you induce SOS system, what it can happen is that, like we saw in the Ratman experiment, many of the bacteria which is integrated stably inside a bacterial system is activated to form, just like in the pathogenicity island, to take into mature phages or viruses. Now, viral particles will be much more in this case. What does it mean is that it will increase the infectivity, or it will help in the spreading of antibiotic resistance genes to other cells through the bacteriophages. How it is important is that, as I was telling you, that the antibiotics, for example, ampicillin, or related, which inhibits the synthesis of peptidoglycans, what it can happen is that it activates that two component system, and one of the protein what we know of is the DPIB system. This system also relates and induce or stop the or response to that system, uh, it can induce SOS response, and it can kick in the stoppage of the cell division itself. Okay, so so far what we have been discussing is that sub-inhibitory levels of antibiotics can induce SOS system, and because, as we talked about, it can help in the lysogenic bacteria phases to come into lytic phases, or the pathogenicity islands can be condensed to form a virus-like particle. It helps in or leading to uh, horizontal gene transfer from one or antibiotic resistance spreading, and it can induce virulence factors spreading. So SOS response is very, very important. So this is what we were talking about the phases. When there is a single system, REC A can break LEX A and induce SOS system. And uh, it can involve, many times, it can involve in the de-repression of bacteriophage proteins, or in another scenario, these REC filament can also help in the phage repressor protein to be clipped just like LEX A. And many of these phage repressors are important for the bacteriophages which are integrated in the host genome to be lying idle. So now the bacteriophage repressor is clear, it's no more repressed, the phage genes are expressed, and the phages can come out as a prophage, and it can help in the spreading of antibiotic resistance genes. So now, why we have been talking so far, and what are the molecular mechanisms? How does it do? How does it uh, induce different varieties of response because of the SOS response? Of course, there are genes or factors which are under the control of LEXA, which help in giving rise to different stages of or different types of SOS response. For example, in this case, what we are looking at is that it can have checkpoint activation, thereby leading to inhibition of the cell division so that the cell gets enough time to repair the damage or complete the cell, complete the replication, enough time is there. Or another way, depending on the cell stage, it can also induce certain types of response just to make the cells inactive and let it wait for the better or compatible situation comes, and then it divide later on. And another way is you can help the bacteria enter into programmed cell death, just like what it has been seen in the case of higher eukaryotes. Bacteria can also enter into programmed cell death if any of the two options, earlier options, are not cannot be performed. In the case of checkpoint, how does this SOS response kicks in this checkpoint protein? There are many factors which are involved in the cell cycle progression checkpoint. What is important here is that one of the first protein that we talked earlier also, cell A. Cell A, what it does is that it is known to inhibit the polymer formation of a protein called FTSG. So FTSG is believed to form a filament in the middle of a dividing cell and helps in the formation of cell wall formation, and thereby leading to separation of the two daughter cells. So now when the SOS kicks in and cell is expressed, the FTHZ polymerization is inhibited, so the daughter cells cannot go to two daughter cells. That is one way to induce or delay the cell separation or cytokinesis. Uh, Daughter cells are not separated. Another way is induction of two polymerases. One is, of course, DNA polymerase 3 is the main polymerase for DNA replication. But in the case of when the SOS response kicks in, when there is a lot of damage on the template DNA, what it helps is that it helps in the induction or expression of two other polymerase, that is DINB, which is the DNA polymerase 4, and uh, polymerase comprised of UMU-D and C called the polymerase 5. Why it is important here is that these polymerases, polymerase 4, which is DINB, and polymerase UMU-D-C, is a error-prone polymerase. So it can bypass the DNA lesions so that DNA synthesis or replication carries on. It doesn't bother whether there is an error. It's just looking at the survival of that cell. So SOS response induce error-prone as well as which can synthesize DNA across a DNA damage or DNA lesion. So these two polymerases are called trans lesion synthesis polymerases, TLS polymerases. So pole 4 and pole 5 proteins are induced and allow replication to continue even when there is a lot of damage on the template DNA. That is one. And another mechanism to lead to the stalling or checkpoint is the one of the factors called YAF-O. YAF-O is very, very important because it affects at the translation level. What it does is that it goes and when SOS response is there, YAF-O is induced, and YAF-O binds the ribosomes which are translating an mRNA. This binding activates the nuclease domain of YAF-O and cleaves the mRNA. So once the mRNA is cleaved, proper proteins are not synthesized. So the cells which need new proteins to be synthesized and go ahead the cell cycle are paused because no more translation is happening. So there are other factors like SID-A, which are seen in other organisms like Colobeta chrysanthus. What it does is that it affects the interacting partners of FTHC, thereby stalling the cell cycle progression. So there are many ways to stall these processes when there is SOS response kicks in. So what we have seen here is that Rekhe filament formation cleaves the legs A, which have been sitting at, which have been replacing the UMU, DC, and DINB YAF locus, and once it is cleaved, the UMU, DC, and DINB are expressed and brings about a lot of different types of regulation. It can be mRNA, cleavage, or translation synthesis. How it is done, the translation synthesis from the UMU-DC, which is called the DNA polymerase 5, is that the single-stranded filament activates the polymerase activity of DNA polymerase 5 and allows synthesis across the DNA damage portion or DNA damage, DNA lesions. So if you look at it, these DNA polymerases, what we talked just now, the DIN-B and Pol5 belongs to the family of Y polymerases, which are called translesion synthesis polymerases, and it inserts nucleotides against the damaged nucleotide and carry on the DNA replication. And what you can see is that the error rate is 1 in 100 to 1 in 10,000. Unlike the DNA polymerase 3, which is the DNA polymerase replication proteins, and, what you can see is that DNA polymerase 3 is here, which gives error rate of 1 in 1 lakh to 1 in 1 crore, or 10 to 10 million bases. So you can see the error rate is much higher when there is a SOS response because of the fact of the error-prone translation synthesis polymerases. So it is hypermutagenate when SOS response is induced. So one way to mutagenize a bacteria or some organism is you can give a heavy dose of DNA damaging agents, thereby leading to the induction of SOS response, and you can get lots of multiples of mutations. And this finding from the bacteria that SOS response, it's very much applicable in higher eukaryotes, including humans. If you look at it, the pol, eta, kappa, all these things are involved in the translation synthesis in humans as well. So this pathway SOS response is very conserved. You can see even in archaea, in eukaryotes, in bacteria, all three kingdoms of life is affected or it's equipped with this SOS response. So I think the basic, when there is a lot of DNA damage or a process which leads to the exposure of single-stranded DNA portions, just like conjugation, can induce SOS response during DNA damage response. And then another way is during antibiotic kind of treatment, which disrupts the cellular integrity, also induces SOS response. So there are many ways to induce SOS response. The fact is that it leads to three outcomes. One is the delay, or make it wait, persisters, make it persisters, or enters into programmed cell death. So with this, we'll stop for today. Next class, very relevant when is a kind of stress. When there is a stress, how do the cell behave? Another topic we'll discuss tomorrow is the stringent response. Stringent response is more of due to starvation of amino acids. We'll discuss that in the next class. So let me know if any one of you have a question. So are plasmids just prophages? No, plasmids are not prophages, but it can circularize and exist like episomes, just like plasmids. But they are not prophages because plasmids cannot infect. Correct? This prophages is more of the phage DNA, circularized phage DNA, or the genomes of phages. It need not be circular. It can be linear as well, depending on the type of the phages. Any other question? Okay, then we'll stop here for today. Mhm.
Thank you, sir.
Yeah, thanks.
Thank you, sir.